Interaction of cells with other cells. Examination of a thin section through a major organ of an animal reveals a complex architecture involving a variety of different types of cells. A little is known about the mechanisms responsible for generating the complex three-dimensional cellular arrangements found within developing organs. It is presumed that this process depends heavily on selective interactions between cells of the same type as well as between cells of a different type. It is presumed that this process depends heavily on selective interactions between cells of the same type as well as between cells of a different type. It is evident that cells can recognize the surface of other cells interacting with some and ignoring others. It is very difficult to study cellular interactions that occur within the inaccessible organs of the developing embryo as tissue formation takes place. Early attempts to learn about cell recognition and adhesion were carried out by removing a developing organ from a chick or amphibian embryo, dissociating the organ's tissues to form a suspension of single cells, and determining the ability of the cells to reaggregate in culture. In experiments in which cells from two different developing organs were dissociated and mixed together, the cells would initially aggregate to form a mixed clump. Over time, however, the cells would move around within the aggregate and eventually sort themselves out so that each cell adhered only to cells of the same type. Once separated into a homogeneous cluster, the embryonic cells would often differentiate into many of the structures they would have formed within an intact embryo. Little was known about the nature of the molecule that mediated cell-cell adhesion until the development of the technique for purifying integral membrane proteins, and more recently for the isolation and cloning of genes that encode these proteins. Dozens of different proteins involved in cell adhesion have now been identified. Different arrays of these molecules on the surface of different types of cells are thought to be responsible for the specific interactions between cells within complex tissues. Four distinct families of integral membrane proteins play a major role in mediating cell adhesion, which are selectins, certain members of the immunoglobulin superfamily, certain members of the integrin family, and catharins. So, ectoderm cells and mesoderm cells are mixed. They are allowed to separate. Then they started developing different structures and they sorted out themselves heart cells and precartilage cells. Experimental demonstration of cell recognition that is happening there. When cells from different parts of the embryo are dissociated and then intermixed, the cells initially aggregate and then sort out by associating with other cells of the same type. The results of such classic experiments are shown here. In this experiment, two regions of an early amphibian embryo, the ectoderm and mesoderm, were dissociated into single cells and combined. At first, the cells form a mixed aggregate, but eventually they sort out. The ectodermal cells, shown in red, move to the outer surface of the aggregate, which is where they would be located in the embryo, and the mesodermal cells, shown in purple, move to the interior, the position they would occupy in the embryo. Both types of cells then differentiate into the types of structures they would normally give rise to. Light microscope showing the results in an experiment in which the precartilage cells from a chick limb are mixed with chick heart ventricle cells. The two types of cells have sorted themselves out of the mixed aggregate, with the heart cells forming a layer on the outside of the precartilage cells. It is proposed that the precartilage cells collect on the center of the aggregate because the cells adhere to one another more strongly than do the cells from the heart. This and other models are discussed later. So we have learned which are the particles responsible for the interaction of cells with other cells, which are the membrane protein, which are the membrane integral membrane proteins, selectins, certain part members of the immunoglobulin superfamily, certain members of the integrin family, and catharins. So first let's learn about selectins. During the 1960s, it was discovered that lymphocytes that had been removed from peripheral lymph nodes, radioactively labeled, are injected back into the body would return to the sites from which they were originally derived, a phenomenon called lymphocyte homing. It was subsequently found that homing could be studied in vitro by allowing lymphocytes to adhere to frozen sections of lymphoid organs. Under these experimental conditions, the lymphocytes would selectively adhere to the endothelial lining of the venules, the smallest veins. Under these experimental conditions, the lymphocytes would selectively adhere to the endothelial lining of the venules. Where are they adhering? Into the endothelial lining of venules of peripheral lymph nodes. Binding of lymphocytes to venules would be blocked by antibodies directed against a specific glycoprotein called L-selectin on the lymphocyte surface. Selectin comprise a family of integral membrane glycoproteins that recognize and bind to a particular arrangement of sugars in the oligosaccharide that project from the surface of other cells. The name of this class of cell surface receptor is derived from the word lectin, a general term for a compound that binds to specific carbohydrate groups. Selectins possess a small cytoplasmic domain, a single membrane spanning domain, and a large extracellular segment that consists of a number of separate modules, including an outermost domain that acts as the lectin. These are three non-selectins: E-selectin, present on the endothelial cells; P-selectin, present on the platelets and endothelial cells; and L-selectin, present on the leukocytes, white blood cells, which are the three positions. E-selectin, present on the endothelial cells; P-selectin on the platelets and endothelial cells; L-selectin on the leukocytes, endothelial cells, platelets, and leukocytes or white blood cells. All three selected recognize a particular grouping of sugars that is found at the ends of the carbohydrate chains of certain complex glycoproteins. Binding of selectins to their carbohydrate ligands require calcium. As a group, selectin mediate transient interactions between circulating leukocytes and vessel walls at sites of inflammation and clotting. Capturing a leukocyte is a challenging task because these cells are flowing through the bloodstream at a considerable rate of speed. Selectins are well suited for this function because the bonds that they form with their ligands become stronger when the interaction is placed under the mechanical stress, as occurs when the leukocyte is being pulled away from a given site on the vessel wall. The role of selectin in inflammation is discussed later in the human perspective, which we will be learning after this. So L-lectin, E-selectin, L-selectin, E-selectin, and P-selectin are the different categories. Then they have structures called lectin-like domain, EGF-like domain, structural domain. Extracellular region is glycoprotein will be there, then P-selectin can be seen, and the remainder of oligosaccharide. Three types of known selectin. All of them recognize and bind to a similar carbohydrate ligand at the ends of oligosaccharide chains on glycoproteins, such as one shown in the figure. The detailed structure of a carbohydrate ligand, the terminal fucose and sialic acid moieties are particularly important in selectin recognition, and N-acetyl glucosamine moiety is often sulfated.